Hello, this is Mike again. Here comes our second lab demonstration. This time, we'll investigate the antimicrobial effect of a Streptomyces graezius strain, which is derived from the strain discovered by Weckmans and Henry in 1948. Weckman eventually earned a Nobel Prize with the discovery of streptomycin produced by Ascariasis. It's also the first antibiotic ever reported from a bacterium.
Ascariasis exhibit different physiology in different media. For example, on TSA, which is Triptychal soil agar, the colony is brown and rigid. However, when the Ascariasis is grown in the liquid culture, then transferred to the TSA, the colony becomes white. The colony of Ascariasis is firmly attached to the agar and hard to be removed. Here is a colony of E. coli. Compared to Ascariasis, the colony of E. coli can be easily removed by an inoculation loop. In all the media described above, Ascariasis release a scent that is similar to the smell of the soil after rain. This earthy and musty odor is caused by a compound called geosmin, which can be produced by Ascariasis. Here is the chemical structure of geosmin. Human nose is extremely sensitive to geosmin, which might be an indicator of the freshness of a water source for our ancestors.
The first experiment will show the antimicrobial effect of Ascariasis strain on the agar culture.
First, we will inoculate the agar plate with liquid Ascariasis culture. Under an aseptic environment, we transfer 10 microliter of Ascariasis liquid culture to the center of the agar plate. And then we leave it in the room temperature to allow the growth of Ascariasis. After inoculation, Ascariasis colony will be visible after a day, and fully grown after 3 days. When the colony of Ascariasis is ready after 3 days, we will spread E. coli all over the plate. Here's an E. coli culture. To avoid the disturbance to Ascariasis colony, we will use the pouring method instead of the hockey stick to spread the E. coli. To do this, we add 2 ml of E. coli culture onto the agar plate. Then we tilt the plate to allow the liquid culture cover the whole plate. After this is done, we leave the plate in room temperature to dry and allow E. coli to grow. Here is the result of our first experiment on two different agar media. Interestingly, we found the inhibition zone to be different between ascaricis on TSA and ascaricis on neutron agar. This suggests that the amount of streptomycin produced by ascaricis in TSA is much lower than it is in neutron agar. One of the key components in neutron agar that TSA doesn't have is meat extract. There are a few publications investigating the factors determining the amount of streptomycin produced by ascaricis, including the media composition, temperature, and growth phase. If you are interested in this topic, please read over them.
The second experiment will attempt to show antimicrobial effect of ascaricis strain in the liquid culture. Previously, soil sample has been found to have an antimicrobial effect. In this experiment, we will determine whether the antimicrobial effect is from the chemical or streptomyces in the soil.
First, we collect some soil sample downstairs of Hoyot lab. Then, we use autoclave to eliminate all the microbes in the soil sample. We add 2 grams of autoclaved soil sample to 2 flasks. In the first flask, we add 10 ml of GMP media. In the second flask, we add 5 ml of GMP media and 5 ml of ascaricis culture in GMP media. The total volume is also 10 ml. Then we inoculate both flasks with a very small amount of E. coli culture. We leave 2 flasks in shaker at 28 degrees Celsius. Eight hours later, we will use standard plate count to estimate the CFU of E. coli in the flask. We first take 1 ml of sample from each flask. We first briefly centrifuge the sample to eliminate soils in the sample. We also use 5-micron pore size filter to minimize soil particles and excrescence in the samples. Then, we make a series of tenfold dilutions of the sample. To do this, we add 90 microliter stereo media to each well. We add 10 microliter samples to the first well, then mix and transfer 10 microliter from the first well to the second well. We repeat this dilution procedure for 6 wells. Plate all dilutions on an LB agar plate, and leave the plate in 37 degrees Celsius incubator overnight. Next morning, we will see the result. If the chemical in soil is responsible for the antimicrobial effect, we will not see the growth of E. coli in both flasks. If the ascariasis is responsible for the antimicrobial effect in soil, we will see the growth of E. coli in ascariasis minus flask, but not the ascariasis plus flask. We found that the growth of E. coli in ascariasis minus flask is much better than it in the ascariasis plus one, indicating that ascariasis is responsible for the antimicrobial effect in the soil. Note that in the first picture, the sample is not filtered, so you will see small white colonies in the ascariasis plus sample. In the second one, ascariasis is filtered out.
In this lab demonstration, we observed the antimicrobial effect of ascariasis. We have found that the amount of streptomycin produced by ascariasis depends a lot on media composition. We have also found that the chemical in the soil does not have much antimicrobial effect, but the ascariasis does. I enjoy doing those experiments as well as the smell of ascariasis. See you next time.